Hello, my name is Buckland. Delighted to be able to join you today, present a masterclass on really with a technology focus for vaccine development. And it's an honor to be part of this 2021 ISV Annual Virtual Congress. I'll be talking to you today about the remarkable progress related to COVID vaccines, and with a focus on the technology aspects. And I'm doing this from the perspective of my current roles. I am a visiting professor at University College London. I have a consulting company, BiologicB LLC. And also, I'm the executive director of Nimble. So we can all be thankful for these vaccines. It's really helped to change our lives. Will continue to be important. My own prediction is we're going to be seeing this for very many years to come in terms of ongoing need to develop improved versions of the vaccines for COVID. Now, this story began in terms of the technology development in January 2020, and that's when the sequence was published. And so really the remarkable progress that has been made is that a group working, many of the groups working to develop a COVID vaccine actually do not need to work with the virus. And this is revolutionary, but they simply need the sequence of the virus. And so the race began in January 2020 when the group in China published the sequence. And what people had previously learned through work on coronavirus is that the spike protein would probably be an excellent target for a vaccine. And so there was a convergence of activity with the spike protein being the main target. But the interesting thing is that groups took many different paths to get there. And so we see the introduction of messenger RNA technology, Pfizer working with BioNTech and also Moderna. We saw work on self-replicating mRNA, ongoing work with DNA from Inovio, using electroporation to deliver the DNA. Again, all of this is for the spike protein. The work at Oxford University, which is really quite remarkable, Sarah Gilbert and her team developed a replicating deficient adenovirus, and they teamed up with Astrazeneca. And in parallel, work at J&J, again using replicating deficient adenovirus as the delivery system for the DNA coding for spike protein. We saw ongoing development, really the baculovirus expression system with insect cell culture or moth cells. And that's really to make the protein itself. The insect cell culture system can be very fast, and I'll illustrate that. It was originally developed, this platform originally developed for influenza. And then other activities such as making the RBD domain of the spike protein, for example, BiologicB in India, and so on and so forth. So great variety of approaches. The one common theme is to make the spike protein. But what was really significant in 2020 is we saw the emergence to center stage of new vaccine technologies. And this is really both the messenger RNA, which was really very new as a way of delivering the spike protein, and also ongoing work. This has been around longer, much longer as a platform, the replicating deficient adenovirus. When I was at Merck, we worked on this to develop an HIV vaccine candidate. So it's been around, and we've seen different variations. The Oxford University group, led by Sarah Gilbert, did remarkable work to put forward a vaccine candidate and teamed up with Astrazeneca. And then in parallel, work at J&J and also in Russia with the Sputnik vaccine. So when we look at messenger RNA, I mean, there's one thing to make the messenger RNA, but we also, groups working on this also have to consider the whole supply chain. I mean, there's different components which are quite specialized, including specialized enzymes. And if someone is the process development engineer or scientist responsible for making material for the clinic, you not only have to make the messenger RNA from a DNA template, but you also need to supply all these other reagents, including specialized enzymes like RNA polymerase, DNAs to degrade the DNA template. You need microbially made phage polymerases and capping enzymes. So from a process development point of view, you need to work on this, on all these different activities. And the supply chain aspects are also important, as well as the analytics. You need analytical assays, and you need to get the results quickly so you can proceed to the next step. And the great thing about the messenger RNA platform is the base components are generally identical across development candidates in terms of the formulation. So only the coding region really needs to change. And that is really tremendous advantage for this platform in terms of speed. And this was obtained from the BioNTech website, but it's on how messenger RNA-based vaccines work. You see the vaccine enters the cell. Messenger RNA is released. Antigen-presenting cells recognize the messenger RNA, and immunogens are built. And then the antigen-presenting cells present immunogens on the surface of the cell, resulting in T helper cell activation, as well as B cell activation. And so it's really, the challenge has always been this, the concept of messenger RNA delivery has been around for some time. The challenge has always been delivery of the messenger RNA so that it doesn't get degraded, and also so that it works well in us, in humans. So a lot of work has been done on the formulation. This is an article taken from an article in Frontiers in Chemistry, and interesting how the solvent polarity can be changed for the lipid-based nanoparticle. And so the general approach with messenger RNA seems to be to encapsulate in a lipid-based nanoparticle And by changing the solvent polarity, you can indeed change the way the lipid nanoparticle forms. And this is illustrated in this slide. Now, another approach to deliver the spike protein nucleotides that deliver using a virus vector vaccine, such as replicating deficient adenovirus. And this is where live attenuated virus is used as a vehicle to deliver DNA to patient cells. And the antigens are produced by the patient cells and exposed to the immune system. This can induce both humoral and cellular immune response in absence of an adjuvant. And the virus can be propagated in mammalian or insect cells. The challenge is the purification and stabilization of a live virus vaccine, choice of a suitable cell bank, and reproducibility of a complex biological process. So these lead to the process development challenges. And so most this audience will be familiar with the progress being made by the Oxford University group. They worked with Astrazeneca, teamed up with Astrazeneca. And in parallel and separately, a team based at Johnson & Johnson using previously developed technology from Crucell have developed vaccine candidates and taken these through various clinical studies to getting emergency approval use for the pandemic. So the adenovirus is a promising vaccine vector. It's efficient at delivering DNA to target cells. It's a large capacity for incorporation of the DNA expression cassettes and low potential for oncogenesis because they do not insert their genome into the host DNA. So an example of this was developed actually originally working, some of the early work was developed at Kairos, and they use this to develop an adenovirus platform technology. And some of the details, this was taken from a publication by Kairos. They used chimp adenovectors. They chose that because there's no pre-existing immunity in humans to chimp adenovectors. It had been well tolerated in over 500 subjects. You get a T cell response, so that's potentially very important. Kairos developed their own cell line, ProCell 92. It's a proprietary cell line, which went into the clinic third quarter 2012, and then they had a GMP manufacturing set up. And so why adeno? It seems to be helpful in terms of developing a strong T cell response. And you get a combined antibody and T cell response. And it's proven to be safe and highly immunogenic. So years ago, we worked on this at Merck, really geared towards developing an HIV vaccine at the time. This was back in the early 2000s. We published this really nice work on purification using selective precipitation of cellular DNA, and point you towards this publication which some very nice work by Anne Lee and her group. Now, some of the process challenges working with a virus is to some extent you need a statistician, because the assays are not very precise. So is 4.5 times 10 to the fifth the same number as 1.1 times 10 to the sixth, or are the assays with enough replicates that you can tell the difference? But the reason I mention this is when you do experiments, the question is the experiment sufficiently powered, or are you working in the region of assay variability? Are there enough replicates in the assay to get a reasonable number? But the really big question is to be, you have to be careful about drawing conclusions, because your experiment may not be sufficiently powered. Some of the cell line questions: is it tumorigenic, oncogenic? Testing for adventitious agents. Can it be grown in suspension culture? Can you use a serum-free medium? Is the cell line stable over a broad number of passages? So these are some of the practical questions. And then the virus: is the strain stable? What method do you use for quantification? What is the accuracy of the assay? Does the virus tend to aggregate? At what concentration will it aggregate? And then the filling model. And that is, if you want a 2-year shelf life of your vaccine, and if the virus is not very stable, you may see, and this is purely as an example, you may see a decline from 10 to the 8th at the beginning of the 2 years to 10 to the 7th. And the big clinical question is it efficacious at the 10 to the 7th number? And is it safe at which is at the end of expiry? And is it safe at the beginning when the dose is freshly made at 10 to the 8th? So these are the clinical questions that have to be addressed. So there's a nice article in the New York Times about how the Oxford Astrazeneca vaccine works, the adenovirus entering the cell, and really it's really the DNA coding for the spike protein. Now, another approach altogether is to use a technology that was initially developed for influenza vaccines, and this baculovirus expression system was chosen, and work was done by Novavax and also by Protein Sciences, which was subsequently acquired by Sanofi. But using the baculovirus expression system, because it can be very quickly changed from one product candidate to another. So it was especially useful for influenza. And Protein Sciences, and I was fortunate to be involved with this development, working with Manon Cox and her team at Protein Sciences on flu-block vaccine. And this was indeed licensed by the FDA in January 2013 with considerable help from BARDA in terms of BARDA funding. But it's a very good solution for the pandemic, and that's why BARDA was attracted to this. The baculovirus expression vector system, the great thing about it is the cell culture part stays the same. You always grow up the same cell line, and it's the baculovirus expression vector system which changes, and you can do that relatively quickly. So it's actually a very useful technology for pandemics, and Novavax are demonstrating that there was some great progress in the clinic in terms of developing a vaccine candidate based on this technology. And this is illustrated here. In the case of protein sciences, this is to make the HA protein as the candidate for influenza vaccine, and illustrating how the expression of the protein, it forms these rosettes, and these are subsequently purified using a pretty much standard biotech type process, chromatography and so forth, extraction, and then it's finally simple formulation with PBS. But these rosettes are preserved, and this presumably is what makes the protein immunogenic. And the HA hemagglutinin is illustrated here. It's a complex protein. It does seem like this expression system is very good for these very complicated membrane-bound proteins such as the HA protein. And this simply illustrates the major steps involved. Really, with the thought being that you could adapt cell culture capacity used for making antibodies you could adapt this to a pandemic vaccine. And in theory, and actually we did this case, we actually worked on this at Protein Sciences to make for H7N9 influenza. And this was the timeline in very, very rapidly able to express a new protein using this system. So in principle, this is a good approach for a pandemic situation. And so when we look at the overall progress, it's really amazing how this has evolved. And so exciting new vaccine platforms have become center stage. And as we all know, the messenger RNA, the Pfizer BioNTech platform has given great clinical data, better than anyone expected, and as good as one dreamed. And the same with the Moderna vaccine. It's really amazing. And those of us who received the vaccine, I hope everyone watching this has received the vaccine, just amazingly grateful to get this protection. And as I actually got the Moderna vaccine, and just watching it go into my arm, thinking about all the progress, all the science that went into that small dose of vaccine, and thinking about the messenger RNA converting to the spike protein, stimulating an immune response. It's really, really, really remarkable. And people working on this should be very, very proud of what you did. And the same with the replicating deficient adenovirus. I mean, the messenger RNA kind of scooped things because the clinical data was so good. But the replicating deficient adeno has also been remarkably good in terms of performance. And Novavax are demonstrating the actual protein can also be used. So very exciting times, really quite extraordinary. So I'm going to close by talking a little bit, excuse me, about nimble, which is the National Institute for Innovation in Manufacturing Biopharmaceuticals. Yeah, I helped work with Calvin Lee, who's the director of nimble and really driving force behind nimble. Over 5 years ago, we put together a proposal, or I helped put together a proposal to get funding for this, which was indeed approved, and the funding came through the Department of Commerce in the US via NIST, NIST. So Nimble is a public-private partnership, started just about 4.5 years ago, funded through NIST. It involves the FDA, involves large and small companies, involves academic institutions, and nonprofits, as well as states. And it's dedicated to advancing biopharmaceutical manufacturing innovation and workforce development. So we've been working on this. I'm the executive director. Kelvin Lee is the director and really the driving force behind Nimble. And we have 62 academic institutions as members, 65 industry members, including most of the major companies, nearly all of them actually, that make vaccines, and 31 other entities like nonprofits and so forth. So we've built all this over the last 4.5 years. We have funding for another 5 years, for which we're very grateful. But also through the recent American Rescue Plan, which was signed into law by President Biden, there's $83 million has been awarded to Nimble for coronavirus response projects, both for current pandemic, but also preparing for future pandemics related to coronavirus. And part of this 83 million includes significant funding to establish a vaccine analytics center of excellence at Nimble. And so we've just started working on this last part. So we're very excited to be doing this, and we'll be reaching out to you in the vaccine community, because we want this to be useful to companies working to develop vaccines. The thought being that if you can characterize a vaccine better, then it makes it simpler to introduce process improvements. It allows the development of better formulations. Once you know what are the critical assays and what are the critical potency assays, it allows the development of better temperature stable formulations, and also allows a company to move facilities from one geographic location to another, or to go up in scale. And so we're excited about this. But I do want to close by thanking everyone working on vaccines. You've really done an amazing job, these companies. We're very grateful. And clearly, this is an ongoing need.